Hello, and welcome to the CAP Today webinar for Tuesday, August 13th. I'm Bob McGonigal, the publisher of CAP Today, and I'll be your host and moderate a question and answer session after our formal presentation. Today's webinar is entitled, Using Rapid NGS Testing for Trial Assignment in the NCI Precision Medicine Initiative in AML and MDS, known as myeloMATCH. And this very critical and timely webinar is made possible by a special educational grant from Thermo Fisher Scientific. We want to acknowledge this important support and thank Thermo Fisher for its help in bringing you this very important webinar today. Today we have a single speaker who's returning in this series of CAP Today webinars, and that delights me. I'll say a bit more about him in due course, but our speaker is Dr. Jerald Radich of the University of Washington School of Medicine and the Fred Hutchinson Cancer Research Center in Seattle. But before we begin, let me just go through, as usual, a few housekeeping tips to optimize your enjoyment of today. First, we suggest that you refresh your browser. We do have live aid available throughout today's presentation. You can see in the bottom right-hand corner of your web scene, your website, 858-345-5916. We have live help available throughout the hour if you have any technical problem with website or audio. You'll see below the slide deck a section labeled Q&A, and while this is primarily for your questions for our speaker and your comments on the topic, you can also type in technical notes then or any problems that you have, and we'll attend to those very quickly and resolve them. And I wanted to let you also know we plan to post this webinar in its full text and audio in about a week or 10 days following today's presentation. You should also know you'll likely get some follow-up email from ourselves and our sponsors, so be alert and be aware. Finally, it's very important that you all understand and note that CAP Today does not endorse any products or services that may be mentioned in today's webinar. Any remarks of mine are purely personal. They are not to be taken as the policy of the CAP Today or, much less, the College of American Pathologists. And so with that, we're going to go ahead and begin. And once again, I will read our slide here. You see the beautiful seaplane there and lovely photo with the Fred Hutch in the left-hand corner of the slide. Using rapid NGS testing for trial assignment in the NCI Precision Medicine Initiative in AML and MDS, myeloMATCH. And I want to read to you some disclosures. I think it's very important. Thermo Fisher Scientific and its affiliates are not endorsing, recommending, or promoting any use or application of Thermo Fisher Scientific products presented by third parties during this seminar. Information and materials presented or provided by third parties are provided as is and without warranty of any kind, including regarding intellectual property rights and reported results. Parties presenting images, text, and materials represent they have the rights to do so. Our speaker was provided with an honorarium from Thermo Fisher Scientific for this presentation. And speaking of our speaker, I'm going to turn to him now. Dr. Jerald Radich, of course, is a professor at the University of Washington School of Medicine, a very distinguished oncologist. He's a member of the Clinical Research Division, and he's the Enslein Endowed Chair at the Fred Hutchinson Cancer Research Center in Seattle, one of our very most distinguished institutes in the whole country, and indeed the whole of Western medicine and this hemisphere. And with that, I'm going to ask Dr. Radich to begin his presentation. Dr. Radich.
Good morning, everyone. Jerry here from Seattle. Greetings. Thanks for being here. First and foremost, we have in the chat there my email. So if there's any questions you want to follow up with, any ideas, any collaborations, any technical issues, that's a good way to get a hold of me. So what I'm going to do today is really a talk in 3 parts. The first is going to be talking about the myeloMATCH program that we've been developing with the NCI for 5 years now and has now just started activation across the country. And that's kind of sensational. Next, we'll talk about sort of something aspirational, using next-generation sequencing as a marker of molecular residual disease and how that might impact how we treat patients. And then lastly, something that's inspirational, how we can basically take this kind of technology and really get to global audiences and try to share the benefits of modern molecular medicine with the rest of the world. So to start this off, just to kind of show us where we have been and where we want to go, and we're showing here the tale of 2 leukemias. And so on top, this is SEER data from the US. So on top here is the rate of new cases since 1992 to 2019 of CML. It's remained fairly stable, as you can see. But the bottom here, this is actually death rates. You can see it was stable here for a long time, and then suddenly it dropped dramatically. And basically that's because we developed the tyrosine kinase inhibitor, Gleevec, and now the 2nd generation drugs. And it just shows really the promise of so-called precision medicine, where we know a molecular target and we can design a drug to that and impact survival. So in CML, we're sort of in a unique situation in that we have really one molecular lesion, BCR-ABL, that drives the disease. That then can serve as both a target for drug therapy and serve as a marker of disease burden, so that in almost all of the FDA trials for the 2nd generation drugs, the BCR-ABL, as determined by peripheral blood mRNA testing, is really the endpoint of the trial as a sensitive marker of the disease burden. So the question is, can we bring that kind of precision medicine to other diseases? And the one that we're going to target here is AML. So in AML, as opposed to CML, you can see the incidence rate might be going up, but survival rates from 1992 to 2020 basically are exactly the same for the death rates. And if you actually push this back to where I got started about 3 decades ago, it really hasn't budged at all. So for all the work we've done in defining molecular lesions, for all the new drugs that we're coming out with, so far we haven't budged on the death rate of AML really at all. And so what the NCI is doing with the myeloMATCH initiative is trying to see whether we can actually bring modern molecular methods, tie that into modern drug development of small molecule agents, and really try to see what we saw in CML and AML. So you've seen this before. This is looking at the mutational heterogeneity in AML. This looks busy, but compared to solid tumors, AML is a pretty clean space. There's really about 25, 30 mutations that cover most of the waterfront of mutations. And realistically, most patients have, on the average, about 3 to 5 mutations. So it's as opposed to solid tumors where you might have, 20 mutations. These can be banked into various functional groups, which I outlined here. And these kind of circus plots show that the association of one type of mutation with another isn't random, that there are some that are quite common, like here's DMT3 with NPM1, NPM1 with FLT3, et cetera. And so there's not an infinite number of a variety of mutations or how they coexist with each other. So it's a potentially a space to go after as far as defining disease subsets and defining clinical trials designed for specific mutational targets. Classification schemes in AML are sort of a cottage industry. This is sort of some of the newest ones based on patient clusters by molecular subgroups, low risk versus high risk. So those are things that we can define at diagnosis. The question is then, if we use those molecular underpinnings and we have specific trials to specific subtypes with specific agents that will target those, can we basically make AML the new CML? And that's really what the myeloMATCH is about. So this is what I used to think of precision medicine. A patient would come in sick, you generate some data, cytogenetic molecular data, you pick a drug, and then the patient would be well. And that's when I was kind of, very naive about how much impact or how much work this would have to do. And I naively thought that most of the data or most of the hard work would be here making the data. Our 5 years of trying to get myeloMATCH together kind of shows how long that was. So the myeloMATCH acronym is NCI Myeloid NGS Molecular Analysis and Therapy Choice. That's the way myeloMATCH comes from. And there's kind of 3 particular features. One is that we are really going to use genetically different protocol assignments. So cytogenetics for rough risk groups, molecular lesions to define specific drug trials that they'll go on. As opposed to most phase 3 or most NCI efforts with the cooperative groups in the past, which have been predominantly phase 3 trials, these will be predominantly phase 2 trials, randomized phase 2 trials. And as opposed to the traditional way of using overall survival as endpoints, which takes 5 to 10 years, the myeloMATCH suite of studies will be predominantly minimal residual disease driven right now, with flow cytometry being the endpoint. So as opposed to 5, 10-year trials, these will all be trials where we'll make a decision about whether the trial is a success or not, really for some trials at the end of induction, for some trials at the end of a consolidation regimen. So really rapid trials that are really meant on drug discovery. So that if you do find something that has a strong signal, you can then fold that into a phase 3 trial. So we're really trying to move the bar here and bringing up new drugs and quickly identifying whether they're going to work or not. So there's many essential components to this, and this is, again, taken 5 years, a Herculean effort. There's the IT team, which is immensely difficult, and they've had to actually build this from scratch. Because you imagine with all the U.S. intergroups, we're going to have hundreds of potential sites that can enroll to these trials across the U.S. So we're going to have to track that enrollment. We're going to have to track the samples as they go to the centralized labs, and I'll be talking about that for a minute. We have to integrate all the clinical information and lab data for a treatment assignment, and then we have to follow those patients. So that's been a huge informatic undertaking. We have a specific team that called the agents team that goes out to pharma to look for new agents to specific drug targets. So you have to do that, then you have to find the partners, you have to drill contracts. This is hundreds of hours of work to try to secure pharmaceutical involvement in supporting these trials. There's a laboratory team, which we'll talk about, which is basically developing cyto, fish, flow, next-generation sequencing, validation assays, all in the CLIA setting, and working with the FDA to basically have an IDE so that we can use these type of methods to use drugs that aren't yet approved. And then there's a trial team, which basically integrates all these labs and tests into new trial design. So this is how it kind of lays out. A patient will come in to a newly diagnosed AML patient or MDS patient will come into any of the sites, and then they get multiple samples drawn. They'll have one sample drawn to the local pathologist who will be making the diagnosis of AML or not. We can't use this the FDA has made it clear that we cannot make a call of AML or MDS basically just on cytogenetics or molecular. We have to have the local pathologist to say that's what it is. And then we'll have samples sent to various places. Flow cytometry will be done at Children's by Brentwood. Cytogenetics will be done here at the Fred Cancer Research Center. And then the molecular analysis will either be done at MOCA at Frederick's lab at the NCI or by our lab at the Fred Hutch, depending on what day of the week is, because they don't work on the weekends and our CLIA lab works on the weekends. And unfortunately, patients come in on Thursdays and Fridays. They will have a large amount of samples that will be sent to nationwide suppositories for future work. We have various what we call tiers, and the way to conceptually think about these tiers is it's based on how much disease people have at the time of diagnosis. So our first trials that are opening up now are Tier 1 treatment trials. So they're all newly diagnosed patients. We have subgets of older AML and younger AML. And these are patients with high disease burden, just at diagnosis. The first reassessment will be done after, for instance, consolidation by flow cytometry. If those patients are negative by flow, they keep going on trial. If they're positive, they will then go to these treatment tier 2 trials, which are essentially MRD eraser trials, where you have patients who have residual disease and you build a new trial to basically attack that residual disease and make them negative. For patients who are appropriate to go on to transplant or who the drugs failed them in tier 2, we'll have a suite of transplantation and cellular therapy trials. And then the tier 4 trials will be something I'll show shortly in this, is kind of using really, really sensitive next-generation sequencing techniques, mostly by duplexing sequence techniques where you can detect potentially VAFs down to 1 in a million, and seeing whether there are leftover disease burden at that point, and then attacking those with specific drugs. So right now, the ones that are rolling out are up on tier 1, tier 2, 3s, and 4 are coming to follow. So the key thing here is that we were told by our clinical colleagues that both for patient care and for study design, they would like to give these novel therapeutics at the get-go. So not have a strategy where you do standard induction therapy and then based on the molecular stuff you get back in a few weeks, adding on drug A or drug B, but rather knowing right up front and then initially treating them with your targeted therapeutic agent. To do that, we need really rapid assessment, and so we are going to be doing cytogenetics, FISH, and the myeloid assay I'll talk about, and flow, all within 72 hours. So we will be making these calls, and we have calls every day where a panel of us are on to review the data that's coming in. So once we get the sample in the door and all the information at the central labs, we'll be making a call within 72 hours of what protocol they'll be going on. So that's kind of revolutionary. And here's what's doing what. Cytogenetics is being done by Min Fang here, flow by Brent, again, gene sequencing by MoCA and our lab. And then for these integrated studies, duplex-seq is going to be done at twin strands and probably ourselves. And then there's sort of more research things of single-cell omics and RNA-seq that we'll be doing here on, but that'll be on a research basis, not really a clinical trial assessment. Now, how we're doing this for the sequencing is by using the Thermo Fisher Genexus system, which is extremely rapid system. It's chipped-based, so as you recall how the ion torrent works by basically doing calling by synthesis, where you're looking at pH changes, so it's sort of the world's fastest, most expensive pH meter. And so this system gives you automatic library preparation, template preparation with isothermal amplification for a few rounds, and then the semiconductor pH censoring sequencing. So that allows us to go, once we get the DNA, RNA available, from putting it in the machine and getting the answer out, really for the actual analysis in the machine, about 30 hours, and then you've got the prep up front. So we can get everything done again in 48 to 72 hours. So the myeloid assay that we're using can do both RNA and DNA. There's 1,600 DNA hotspots, 800 RNA fusions and isoforms. These are the sample inputs, not bad at all. You roughly get about 2 million DNA reads and 300 to 500 RNA reads, and they're targeting basically AML, MDS, and NPM panel. So these are the various hotspots genes, the full genes you can look at, the driver genes, and then there's some expression stuff that we have in some control genes. Now, to go to the FDA required an enormous amount of work to go to the IDE. So the IDE that we submitted to the FDA was 1,400 pages of data. That's right, 1,400 pages for those of you who just fell to the floor. And so this was an immense amount of work done by our lab in MoCA. Cecilia Young was the molecular pathologist who headed up the work here, so all hats off to her. And this is sort of a very, very high-level summary of the data that we sent to the FDA. We did 163 unique samples of patients and healthy donors. We had to do proof of blood, we had to do bone marrow. We did cell lines as well and contrived samples. These are the numbers of the DNA and RNA that we submitted for this. Long and short is sensitivity of the assay was basically 98.6 at one center, 98.9 at the other center. Specificities were 100% both at MoCA and at the Fred Hutch. Accuracies, fantastic. And the positive percent agreement and negative percent agreement are, again, fantastic. This is some of the nuts and bolts data here. I won't run through all of this because there's too much data. If any of you want to see any of this data in detail, I promise not to send you the full 1,400 pages. But if there are specific things you want to know, we will be happy to provide that to them. But the long and short is that we had to prove that we can do this in time on contrived samples and patient samples. We did that, and we basically certainly passed the bar as far as the FDA is going, as far as all of these quality criteria that we have to meet. There's a few issues of limit of blank. We had no false positives. Limit of reporting, we're going to be using for this trial 5%, except for FLT3 ITDs, we'll do the 1% for that, for the VAF. And limit of detection, we're going to be calling for as far as the FDA in the trial at 5%. We can go lower than that, but I think this is the level that we're comfortable calling. There are a few limitations. There are a few blacklisted variants that these are areas of basically unclear pathology and have a little bit of variation in read calls that makes them very difficult to interpret. Expected run failures, most of these are from either problems with uniformity across the various targets, or sometimes no template controls being a little higher than you want. And we're at about 7.5% run failures, but if we repeat, we still make the 72 criteria. One thing that brings up is FLT3 ITDs. As you know, these are very challenging to sequence because of the informatic issue, right, of telling, depending on how big your sequence reads is versus how big the ITD is. We tested this with a number of centers. We sent around 70 samples with ITDs that we had information on, where we assumed that truth was from capillary electrophoresis. And we blindly sent these to 5 different labs at different technologies to see how they would do, and that's a paper in itself that we're writing up. But for the Thermo Fisher technologies, basically we missed 3 very, very low VAFs, and these are VAFs that we wouldn't even call anyway. But the more critical thing is the size of the ITD. And for this assay, for the Thermo Fisher assay, we can detect between 21 and 117 base pairs. Now, the ITDs in FLT3 go to up to 300 base pairs, but if you look at our data, and that's as published out there, if you put the cutoff at roughly 120, you will miss cases. You'll miss somewhere probably between 4 to 8% of FLT3 ITDs. But the FDA said that was okay. That was the cost of business. They're not requiring us to do any reflex testing for those people who are FLT3 negative. So that's just what we're going to have to deal with the assay. This is sort of kind of what the molecular report comes out as. It just kind of identifies, like most of these clinical reports, it's a nice, grayscale color. Tells you what the markers you identify, tells you, if there's any comment with low coverage, and also gives you, on subsequent pages, basically a description of the biomarkers and all the assay details and references, and probably much more detail than most centers or clinicians will want to deal with. Let's go. Try and get the next one, sorry. So this is just the conclusion of this, of how this assay works. And I gotta say, when I said it was sensational, I think it really is sensational about the sensitivity and specificity. How we'll do this is we have daily calls at 10 a.m. I'm not on the call today, but there's a panel of clinicians, IT people, and the lab people ourselves who go all over the calls from the previous days and make the trial assignments. And so that's done on a daily basis. It's really quite an amazing effort to put this all together. This is a Gantt chart that's a little bit outdated now. We have right now 4 trials that are up and running. We have up to 70 centers that are activated. We have more daily. We have one trial that's the master screening and reassessment protocol, which basically allows us to do all of this testing. And then we have specific clinical trials. And right now we have a trial in younger patients, standard risk, a patient in younger high-risk patients, and a patient in younger intermediate-risk patients. We have 8 trials that are in various ways of going. We think at steady state, we'll have about 10 trials open at any one time, and we're hoping to accrue about 2,000 new patients a year is what our hope is. So that's the first part, the sensational stuff. Now I want to talk about the aspirational stuff. As many of you know, most of what my work has been done on is minimal residual disease, both defining assays, moving the clinical trials in CML space and in the AML and ALS space. And the question is right now, we are doing the MRD and AML by flow, but can we basically get to molecular markers, and will those be more sensitive to flow? And that's what this part of this talk is about. So this is looking at a large meta-analysis that was done by Dr. Nick Short and others, looking at survival and based on MRD status at the end of induction therapy or consolidation therapy. And this is, again, looking at MRD negative and MRD positive, and the relative risk of survival or relapse is about 2.5-fold in most of these studies. It doesn't make much difference on these kind of curve diagrams, whether you're doing it by next-generation sequencing or by flow cytometry. You get relatively the same relative risk. Same with if you're doing induction or after consolidation, or if you're in pediatric or adults. MRD is bad, and it's bad at the end of chemotherapy. It's bad prior to transplantation, et cetera. Now, one of the questions is, from a regulatory standpoint, if you were going to use MRD as something for drug development, is these two curves are too close together. What you would really rather have is assays, because you've got 40% of patients who are MRD negative who still die or relapse, and you still have these people who are MRD positive who somehow don't relapse. So the question is can these next generation sequencing technologies maybe pull these curves apart? And at that point, you'd have a better surrogate for early drug studies. And so how we have chosen to do this is with duplex sequencing. So those of you who don't do duplex sequencing, as you know, the limit of standard next generation sequencing, where you're basically making a library off of one strand, is that there is error in the library development, and that usually happens, about 1 in 1,000 base pairs you'll get an error. If you do it off RNA, it's probably even higher than that. We've done calculations that you'll have a background error made in about 1 out of every 24 transcripts you make into cDNA. So your sensitivity of detecting any relevant mutation is really dependent on that error rate. And so that's why in most next-generation sequencing platforms, you really can't make a call below 1% or so, because after that, it's mostly error. What duplex sequencing does is it actually makes libraries off of both strands, and then you only make a call where there's a complementary base in the exact location. So then your theoretical limit is, if you have 10 to the minus 3 on one strand, it's 10 to the minus 3 times 10 to the minus 3 times the complementary base pair. So the theoretical limit is about 1 in 10 to the 7th. So this is a study we did where we took roughly 70 patients who had a diagnostic and CR sample in one SWOG trial. And we, with twin strands, made a 29-gene panel, and we had flow cytometry done by Brent Wood's lab on all these samples. So we blindly assessed them by sequencing and said, how do they compare? And so I think really the most informative thing is this plot. This is like a 2-by-2 table. And so here, if you look at the ones who are concordant, flow negative and next generation sequencing negative, and look at those who are concordant positive, about 70% of them are concordant, which means about 30% or so are discordant, and most of the discordants are patients who are flow negative and duplex sequencing positive. And if you will read this little bullet down here, most of these patients end up relapsing and dying. So on this analysis, duplex sequencing might look better. We did kind of a pilot on how you would define duplex sequencing positive, and you could do that by taking away the so-called DTA genes. You can do it by taking any mutation at any level. There's a variety of ways of doing this. And since we were really just kind of piloting this to see what would work better, we had no really skin in the game. Flow cytometry in this assay was exactly what you find historically, about 2 to 2.5-fold odds ratio. But the duplex sequencing was a lot better. So you could basically, if you looked at time to relapse, the odds ratio here is 7 compared to 2. And for survival, it's about 5-fold. And here are the curves looking at pure flow side, looking at either relapse-free survival or cumulative incidence of relapse, where positive and negative, and you can see these curves are starting to really splay more apart with duplex sequencing. And so that's very exciting, and we hope to bring this kind of technology and compare it head-to-head in the myeloMATCH program with flow, and if it's better, we'll plan on switching to that, because this might be a very, very fast and quick way of really determining who's going to relapse and who's not going to. So the issue here also in data that I'm showing is once you get to sensitivities of this robust, down to the like 1 in a million, it looks like you can probably exchange bone marrow for peripheral blood. And that's something we're going to be ongoing evaluation. As you know, we've done that in CML, where no one gets a bone marrow anymore. And we hope that this kind of sensitive technology will really kind of change the landscape for patients so that we can really get away from bone marrow to just do peripheral blood. So the last thing I want to do is really take a turn different. But as you can see, this is all going to come together. And this is something that's going to be, I think inspirational to most people. And so this is going to go back to CML at first. This gentleman here is actually a physician, and he is riding on a camel for a couple of days to go to an academic center in Sahara Desert. And the reason he's going to do that is because he thinks he may have CML, and if he gets tested for CML and is positive, he will get free tyrosine kinase for the rest of his life. And that's one of the stories I wanted to talk to you about. CML is really quite interesting that the median age in the Western civilization is 60s, 70s. In the developing world, or lower middle income countries, probably a better way to put it, the disease median is in the 30s and 40s, and they tend to present with more aggressive disease. So it seems to be a little bit different disease. Now, how this happened, and what I'm going to get to eventually is, to go back here first, setting a free drug for life. So there's a nonprofit in Seattle called the MAX Foundation. MAX was a kid with CML who died before the age of tyrosine kinases at age 16 because he was from Argentina. It was before we had large unrelated donor registries, and so he died of this disease. Just to remind everyone, before tyrosine kinases, if you didn't get transplanted, your average lifespan of CML was 7 years. And now with tyrosine kinases, the lifespan is essentially normal. But back to Max. Max's mom, Pat Garcia Gonzalez, was a fisheries student at UW, didn't know anything about CML until Max got it. And after he died, she's also from Argentina. She basically abandoned her fisheries career and first started a basically support system for people in low income countries to learn about CML, and you really couldn't do much about it, but could really, just kind of a support system to help them build networks and basically, deliver the disease. Once tyrosine kinases came around, she approached Novartis and said, asked them for help, and Novartis made an arrangement with her that they would give her a drug for life for any patients that she could identify with CML. Now, I think there was a bit of an empty gesture from Novartis because there was no way to diagnose CML in the rest of the world. And so Pat started on this anyway, and eventually she found me because we shared a mutual friend who had CML, and we started doing testing for her, first in El Salvador and there's other places in South America. But this wasn't sustainable because you had to have patients send samples across the world and stuff. So this photo leads us into our next really kind of interesting aspect. as you remember, after 2001, there was an anthrax attack. These are the envelopes that they came in. This is the Postal Service. And so in response to that, this company, Cepheid, started to make automated systems for developing or for testing for bioterrorism. And so Cepheid started, you probably know them now as testing for infectious diseases. They all started first as a bioterrorism company. Most of the mail done in the U.S. is screened for anthrax by Cepheid Systems. And they came to me and asked if I wanted to get into because they were interested in getting into the cancer space. And so with them, we developed an assay that could do BCR-ABL on peripheral blood. Now, this was a game changer because in the developing world, we had tried to set up labs and the like, and it was just too hard to train technicians and the like. And once we developed this cartridge, it just happened that at that time, the WHO had gotten wind of Cepheid and was putting these machines out everywhere in the global area to test for HIV and TB and the like. And so we kind of partnered on them. And so this allowed us to basically bring this kind of technology to most of the globe. But there are areas that still can't have access to it, the electricity and the like. They were still sending us samples by blood, but that, again, that's unsustainable. This is Jordan and Amy in our lab there. She's a PhD. Jordan is a mission guy who goes around everywhere and teaches around the world about how to do Cepheid stuff. What this World Courier bag is, this was basically a large bag that had dried ice in it and one sample from Africa. That costs $500, which is not sustainable at all. So we decided a few years ago to see if we could use some other way to get samples to us to test. And we came upon, after about 2 years of testing, a type of paper that would actually hold spots and not degrade DNA or RNA too much. As you know, for BCR-ABL, you need to do an RNA test. And so most of the papers didn't work. We finally found one that did. And with that, we found that if you compared samples that were drawn and done by Cepheid fresh here in Adelaide versus samples who were sent to Seattle by snail mail up to 4 months out, you would get the same result. And so from that, we've started a large project where we've done, this is old, we've done thousands and thousands of spots from around the world to diagnose CML. This was our claim to fame in the New York Times. And so this has really made a huge difference because, you know, now Pat is, and Max Foundation is taking care of over 60,000 CML patients around the world. And so one thing we asked was, well, if that's the case, are we really doing these patients any good? This was an analysis done by some colleagues at Boston University looking at 60,000 patients on the CML program. This is their survival up to 10 years. This survival for CML patients in Africa and Asia on this program is identical to the CML survival if you were diagnosed in North America. So this just shows the power of targeted therapy. If you can get drugs to patients somewhere, anywhere in the world, if you can make the diagnosis and get drugs, you can give them the same experience of survival as they would get in North America or in Europe. So why that's important here is that we've now been able to, with these dried blood spots and with the platforms I was discussing earlier today, we can now do whole gene panels for all the myeloid markers I showed you on the Genexus assays from dried blood spots. So this is an analysis. These are basically just maps of corresponding to the number of patients we've done, where we can not only do CML, we can do their mutations, we can do, this is for their ABL mutations and resistance, but we can also do all the myeloid genes, the hundreds of them, that are responsible for resistance in CML, plus all the genes that have potential targeted agencies now that Max and others are trying to move out into the rest of the world. So we can really now start applying these new technologies and really make a huge impact for the care of leukemia across the globe. So I'm going to end with this. This is sort of how we see things should work. At diagnosis, you can use mutation analysis, drive prognosis stratification. You can say drug choice. When patients go into remission by conventional morphology, we can use these assays to predict who's going to go there and who's going to go up and relapse. and perhaps change therapy. We can use these assays as drug development. We can use them as scores for eraser trials. And of course, the wish list for these assays is you'd like them fast, reliable, sensitive, flexible, and cheap. But as we've learned, this is a quote from Jim Jarmusch, the director, to Tom Waits. Fast, cheap, and good, pick two. If it's fast and cheap, it won't be good. If it's cheap and good, it won't be fast. If it's fast and good, it won't be cheap. Fast, cheap, and good, tick, 2 words to live by. And with that, I will open it up to questions. Thanks for your attention.
Jerry, thank you very much. What a wonderful talk, and what a wonderful project you've shared with us for this myeloMATCH. It's clearly an epic journey for you all, and it's going to be fun to see this progress as it goes through. I should tell the audience that we've been working with Jerry to also do some further coverage above and beyond what you've heard today, FORCAP today, and he's going to share some other sources and investigators that have been working here. So hats off to Jerry Radich, and hats off, of course, to Thermo Fisher. It's difficult entirely to know where to start, but let me start with some of the more simpler things. We said that the NCI insisted on local pathologists and their diagnosis. Are those confirmed at the various sites to ensure that they were correct in the first instance, Jerry?
We are relying on the local pathologist to be the gold standard for the morphological assessment. They can also do whatever flow and cyto they want to do on site, but they have to send samples as well to our cytogenetics and the like. And then they can integrate those if they want as well. But we are, and the FDA is relying on the local person to make the definitive call that it's myelofibro or AML.
Thank you very much. So obviously, the diagnosing pathologists are absolutely critical to the success of this effort, yep. And it's nice to have them acknowledged as you have here.
Yeah, we can't go anywhere without
following up on, we couldn't do without them. Now, just to follow up on that, for those who are very interested in reporting and reports, is there within this myeloMATCH initiative a workflow to get the molecular cytogenetic reports back to the local treatment team or the diagnosing pathologist so that it can be integrated in the pathology report of the patient?
Yes, yeah, we have a pipeline that when the local center sends us information, there is basically a dashboard that the local site will have where all the information will come back to as soon as it's downloaded.
Thank you very much.
As soon as we push the button and say it's confirmed, that will be sent electronically to the referring site.
Thank you. Thank you very much. Let me repeat your web address or your email address just briefly for all. It's jradich, R-A-D-I-C-H, all one word, at fhcrc.org. That's the Fred Hutch Cancer Research Center.org. And as I said, you're very generously offering yourself for counsel, report, or follow-up, and so I hope this audience can take advantage of that. Let me turn just slightly to the actual work at the University of Washington and your center in clinical practice. What is the role of flow AML MRD testing within clinical practice in your institutions, Jerry? Is it still performed, or are you switching over to NGS and molecular testing?
No, not yet. We're still basically a flow-based institution. The next generation sequencing stuff, I think that most people, I showed you that one study that was with an intense regimen. We've got a study going in non-intensive regimen. In that case, it's a little bit trickier because you never really clear the mutations by when you use a really sensitive technology in people who are getting less intense chemotherapy regimens. So I think that no one yet is quite yet ready to turn over, right? And the other thing is, this rapid stuff we're doing here is for diagnostics. The workflow for MRD level sequencing is much more labor intensive, whereas flow is, same day, right? And so
Right.
I think we've got ways to go until, we can get to the point where the next generation sequencing workflows for MRD is going to supplant flow.
Okay, very good. Thank you for that answer. We also have a question regarding what did you really designate as the gold standard for assay validation in your FDA submission for the IDE status for this work?
Well, that was really an ongoing discussion with the FDA, right? There are no controls right now. There are no controls. We have an initiative with the Foundation of the NIH to develop and validate controls for next generation sequencing. So that's an ongoing work. So what they really required was, I would say that the vast gist of what was done was both reproducibility studies, in your own institution on patient samples. So a patient sample done 7 times on different days or whatever, and then taking those same samples and breaking them so that we could prove that what was done at MoCA and the Fred Hutch jived. And some of that was contrived samples with cell lines, but the FDA was pretty rigorous about wanting patient samples for most of this stuff.
Very good. Thank you. Thank you. We've had several kudos going your direction from people very glad to hear that duplex sequencing is still part of the phase 4 in the myeloMATCH trial. Could you say a little bit more about using duplex sequencing and a little bit behind the scenes as to how that was established and established within the myeloMATCH trial?
Well, the technology, there's various takes on this technology, but the original technology rolled out of Larry Loeb's lab at the University of Washington. And our initial work was with CML and looking for rare mutations in patients who were resistant. But this was the natural rollout. So this, it took a while to develop really develop and validate the actual panel of the 29 genes I noted. And then from that it was really, this whole thing of contracting was done rather secretively. So the contracting company Leidos from the government, they picked all the central labs, including duplex sequencing of twin strands, blindly. So when we had meetings, we couldn't discuss individual labs specifically and the like. They took recommendations by colleagues, NCI and the like, and went away for about a year, and then suddenly just dropped contracts on us. So one Friday afternoon, my grants and contracts officer said, we just got this thing from Leidos about would we want to apply for this thing or not. And so it was all, I couldn't speak to what specifications they actually picked because they never let on, and I don't know, but I know that we're doing it, and I don't know how they picked twin strands, maybe because they were associated with us, but it was rather a mysterious thing. But here we are.
Understood. There you are. Yes. So as always, when we have a lot of lab folks and pathologists on, we're interested in sample requirements. So we have a question here. Your sample requirement slide for this myeloMATCH trial, there's a need for 1 buccal swab. What is the purpose of this specimen, and does it need to be submitted for every AML patient along with other samples?
It's not mandatory. We like it because we anticipate down the line somewhere that with the bank samples we have, that eventually there'll be whole genome sequencing efforts going on, so we need the buccal as a control. That's sort of the anticipation. We're not using them right now. They'll go into storage.
Thank you. Thank you very much.
And if people don't want future whole genome sequencing studies and the like, they can opt out of that in the consent.
Thank you. Thank you very much. of course, we all know that the entire world of healthcare is squeezed for labor and lab staff. In fact, in every nook and cranny of healthcare, people are squeezed for capacity, expertise of up and down the line. But so let me ask you about this. People are very congratulatory to you and your colleagues for this work. But with the samples that may be pouring into your labs, do you see any limit in accepting the number of samples per day or week? And probably out of envy or condolence, what is your lab staff size that handles this work?
Min Fang, the cytogenetics side, she has, I think, about 30 people in her lab, in the cytogenetics lab, and we hired we've fired 5 new people just to take on this because, these samples will have to be fast-tracked. On the molecular side, we have, we've got about 4 techs devoted to doing this, plus people who are sort of, dedicated informatics, mutation calling people. So it's a pretty big staff for sure. How many samples, fortunately, there's not that much leukemia in the world, AML. So, we anticipate maybe at maximum we might get 5 samples in a day, which we can easily add. When we were doing the CML trials for B-Cerebral, there were times where we would get 15 samples in a day. And we've been doing all of the COG AML trials. So, it wasn't unusual for us. And that time we had 3 staff members and we're doing 15 samples a day. So people were busy. And we're developing automated ways to do this. So hopefully those will really kind of, change the workflow a bit and it won't be all manual. But there's no doubt about it that it takes, to move things this fast, the rate limiting step is actual people, right, at this point.
Excellent. And along that line, we have a question about FISH analysis. Is it a mixture of automated methods and manual analysis by your cytogenetic technologists?
Yeah, that's correct. And then they also will be doing CGAT on a select few of patients where the cytogenetics and the FISH isn't clear. They'll be doing CGAT as well. Obviously, that's not going to be a little bit longer than 72 hours.
Yes. Thank you. Now, let me just ask you, clearly, for this to work optimally, you're going to need a fair amount of cooperation with pharmaceutical companies and drug developers. Yes. You mentioned that this can be a bit fraught as you look to sign contracts and agreements. Is there increasing progress and understanding of this, and you're getting, in other words, more help from that entire sector than you might have at the beginning?
I think that it's coming around, right? Because everyone has their own agenda, right? And so how pharmaceutical companies might see how they would like to design a trial, which is often making sure that the control arm has the least efficacy as possible. May not be what the real world wants to see, right? And so we have to there's that issue, but I think the interest in the pharmaceutical companies is best shown by the foundation of the NIH program that I was talking about in MRD and AML. And so how the foundation of NIH works is they sort of pick interesting topics, and then they'll match some funds with industry partners, which have how to come to the table. Usually, for most of those projects, I've been told they usually come up with 6, maybe 5 or 6 industry partners. When we started the MRD for AML, we had to close at 20 partners, about half biotech, half pharma, because everyone sees this as where things have to go, right? And the pharmaceutical companies obviously are really excited because if they get on board and you can really define whether a drug is going to work or not very quickly, that's brilliant for them, right? Not just patients, but for them. The biotech companies, everyone's looking for the next best mousetrap, right? And so they see if their mousetrap is integrated into these trials is golden for them. So we've actually had to stop people from joining into the FNIH because we just couldn't handle all the MTAs, basically.
Okay.
But I think
that's very good.
I think that's an indication, though, of how visible this is, and how there's going to be a push by pharma and industry really to get into this domain.
That sounds good. That's very, very encouraging. I'm delighted to hear that. I'm also, it's a bit old home week to hear about BCR, ABL, and Cepheid. I remember being in John Bishop's office at Cepheid, and he was extraordinarily proud to be presenting that test and explaining it to me on a visit I was in with him. So congratulations again.
Yeah.
A lot of technology behind everything we do.
Absolutely. It's great fun. Great fun.
Good. I'm going to close with one more question. This may be a little recherche, but I know you'll be up to the task, and I hope our audience can appreciate just how much initiatives there are in this field. How are myeloMATCH and other umbrella initiatives, such as Beat AML, similar, different, or complementary? And you can limit your answer to a couple of minutes.
No, yeah. So Beat AML, we some of my best friends are the people who are running the BML, and I just talked to one of them last night about this. So Beat AML is really a fantastic initiative, really looking for signal finding. And so we're using that as sort of a complement of this, so that when Beat AML goes and finds these really, really novel drugs and the like, and are looking at them mostly in phase 1 trials and the like, if they have hits, then they can roll over to myeloMATCH, where we can really scale this to a large, phase 2 trials and bring to bear all of this technology. So we really see Beat AML and myeloMATCH as being kind of an extended arm where one complements the other, because that way Beat AML doesn't have to be sort of, in and of itself. It has all of its great work and funnel into a larger initiative and make everyone happy and get these drugs out there faster.
Thank you. Thank you very much. And with that, I'll give you the last word. I do want to assure some in the audience who arrived a little late, we will archive this talk. Both the slides and the audio will appear in about a week at captodayonline.com. I want to again reiterate the generosity of Jerry Radich by offering you his email address. It's jradich@fhcrc.org. And also, I'm sure your local Thermo Fisher reps will be happy to help you in any way. And so now to sum up, obviously, we're indebted to Jerry Radich. Once again, a wonderful talk. Both aspirational and in the trenches. It's just a perfect view of this area. Very well done. And so thank you, Jerry. Also, of course, I want to thank Thermo Fisher for their sponsorship. These special educational helps really make this all possible. So we're very, very grateful to you. And finally, I want to thank all of you who are on the webcast today. I know you're very busy. We all know how busy we are in healthcare in every nook and cranny. And I want to thank you for taking your time to participate. So to sum up, thanks to Dr. Jerry Radich, thanks to Thermo Fisher, and thanks to all of you. And with that, I'll bring today's webinar to a close.